Okay, so we are going to look at the next chapter, okay, which is on chapter 3, which is on plasma membrane transport. So the slide is not a lot, but I don't know, maybe my explanation will consume much time. Okay, we'll see. Okay, so okay, so these are the learning outcomes, okay, for chapter 3, which is list, yes. Okay, so which is list the components of plasma membrane and their function. The first one, okay, you have to know what are the components of the plasma membrane. So as you know, the plasma membrane, its main component, okay, is the phospholipid, okay, phospholipid molecule which are arranged in bilayers. Okay, so you have phospholipid molecules that are arranged in bilayers, and then you have other components that are embedded in the plasma membrane, such as, for example, you have many proteins that are embedded in the plasma membrane, and this protein will have many or several functions that we are going to look at later, which is for learning outcome number 3, okay, which is you have to know the role of the membrane proteins. So the proteins can have functions such as it can function as enzymes, it can function as the binding site for hormones, okay. It can function as carrier proteins, or it can function as proteins that attach cells together. Macam kita tengok tadi kan, the desmosome, the tight junction, the gap junctions kan. So those junctions are actually proteins that are embedded in the plasma membrane. And then it has if I'm not mistaken, there will be six major role of the membrane proteins. Okay, so the protein that are embedded in the plasma membrane, we call it as the integrins, okay, or the transmembrane proteins. Okay, but there are also another type of protein that are found nearby the plasma membrane. So these protein are not embedded in the membrane. Awak tak faham kan perkataan embedded? Embedded tu maksudnya tertanam lah. Okay, so dia tertanam dalam plasma membrane. Tapi ada juga another type of protein that are found nearby the plasma membrane, which is not embedded. Okay, they are found on the just outside of the plasma membrane, but when I said outside, it is not actually outside of the cell. Okay, it is a protein that is near the plasma membrane but is inside the cell. So let's say, for example, okay, you have here a cell. Okay, so this is the plasma membrane, then you have protein embedded. Okay, so you have another protein that is not embedded. We call okay, this protein, okay, they are found near the plasma membrane, but they are facing the cytoplasmic site. Okay, so nanti kita tengoklah the different types of protein. Number 2, describe the structure of the plasma membrane and the function of each of its component. Okay, so you have to know that the membrane, the structure of the membrane and the function of each of the components. Okay, so apart from having protein, it also has other components such as cholesterol. Okay, so cholesterol that are found in plasma membrane are only found in animal cell, and they are not found in plant cell, which have certain, the presence of the cholesterol will have certain function. Okay, that one we are going to look at later, what is exactly the role of cholesterol that is embedded in the plasma membrane. And then the third one, explain the role of membrane protein, I've explained. And then number 4, explain how membrane structure results in selective permeability. Okay, so that is another role of plasma membrane. Plasma membrane, it controls certain substance to enter and to exit the cell. Okay, So the major component of the plasma membrane, as you know, it is consists of phospholipid molecule. So phospholipid molecule is lipid. Okay, so it means that one of the ability of the plasma membrane, it allows for the diffusion of for example hormones that are made up of lipid, which is steroid. Okay, steroid hormones such as testosterone, estrogen, progesterone, because it is made up of steroid, which is lipid. So this type of hormone can easily diffuse through the plasma membrane to enter into the cell to coordinate the cell function. Okay, and then other substance such as polar substance. Okay, polar substance such as glucose. Glucose, as you know, it is polar. Okay, so for polar substance, they or ions, okay, such as sodium ions, chloride ions, these molecules or ions cannot easily pass through the membrane of the cell. So in order for this molecule or ions to enter into the cell, they have to go through the protein, okay, that are embedded in the plasma membrane. So they have to pass through transport protein. So for transport protein, there are two types, which is carrier protein and also channel protein. Even for water molecule, it is a small molecule. Tapi kita belajar water molecule is polar. Okay, so water can diffuse through the plasma membrane but at a slower rate because due to its polarity. So in order for cell to pass through the plasma membrane quickly, so they have to pass through using channel protein that is specific for water. So channel protein that is specific for water, we call it as the aquaporins. Okay. So and then so that one we are going to look at in detail. What are the factors that causes membrane to be selective, okay, in allowing certain substance to enter and exit the cell. So number 5, explain the various transport mechanism across the membrane. Okay, so substances can enter into cell or exit the cell through either diffusion, okay, diffusion such as tadi apa nama cholesterol, okay, or some other molecules can enter into the cell through passive transport. Diffusion is a type of passive transport, but you have passive and also active. Okay, so for passive you have diffusion and you have the other one, which is facilitated diffusion. Okay, facilitated diffusion, it allows substance to enter into the cell according to concentration gradient from high to low concentration, kan? Concentration gradient. So in order for this substance to enter into the cell, okay, or exit the cell, is through we call it as channel protein. Okay, contohnya channel protein, which is a type of transport protein such as aquaporin lah. Tadi kan yang saya mention aquaporin. So another type of transport mechanism is active transport. Okay, so active transport, as you know, is a mechanism that involves expenditure of energy. Okay, expenditure of energy. So this usually the energy comes from ATP. Tapi nanti kita akan tengok later, the energy doesn't necessarily comes from ATP. Okay, so nanti kita tengok the one that involves a mechanism that involves a type of protein, we call it as co-transporter. Okay, co-transporter. So that one is actually a type of transport protein, active transport that indirectly uses energy from ATP, but directly it involves another type of ion to allows other substance to enter into the cell. So that one kita akan tengok lah. So active transport for the movement of substance across the cell, and then under active transport you also have two other types of transport mechanism across cell, which is endocytosis and also exocytosis. Okay, so endocytosis, exocytosis, if you remember during chapter 2, saya ada sentuh sikit perkataan ni, endocytosis and also exocytosis. So that one we are going to look at later. Okay. Okay, so okay, so that is the LO, okay, learning outcome punya briefing. Okay, so now we are going to look at in detail the for each of the subtopic. Okay, so subtopic 3.1, which is membrane component and its organization. So the plasma membrane, basically the function of it is to separate living cells from its surrounding. Okay, so you have the cell, okay, and then inside the cell you have the nucleus, you have other organelles, and then outside the cell you have the extracellular fluid. Okay, so number 2, control traffic into and out of the cell. So like I've mentioned, so in order for the cell to function or carry out its activity, it has to allow substances to enter and exit the cell. Okay, such as for example, a cell function to produce hormone. Okay, so in order to produce this hormone, hormone or enzyme or other products. Okay, so in order for the cell to carry out its function, it has to allow substance to enter and substance to move out of the cell to carry out its function. Okay, so the membrane, the plasma membrane, will exhibit selective permeability. Only certain molecules, certain ions, okay, can pass through certain cell. Okay. So phospholipids. Phospholipid is the most abundant lipid in the plasma membrane. So as you know, it is to form the phospholipid bilayer. Okay, phospholipid bilayer of the membrane. So without the phospholipid molecule, there will be no cell. Okay. So the property of the phospholipid molecule is that it is amphiphilic. Okay, bukan amphoteric. So amphoteric is referring to what? Amino acid yang kita tengok chapter 1 kan. Amphoteric and amphiphilic is totally different. You have to know which one is which. Okay, amphoteric is when a molecule has both acid and basic group. Kan, it has both amino group and carboxyl group. Okay, that is, it can become acid, it can become base. Okay, amino acid, for example, that is amphoteric. Okay, so but this one is amphiphilic. So amphiphilic means that the molecule can be both at the same time. It has hydrophobic region, it has hydrophilic region. So for example, this is a phospholipid molecule, so you have the hydrophilic heads, okay, and then you have the hydrophobic tail. That is amphiphilic, having both hydrophobic and hydrophilic region. So the another molecule that has this property that is amphiphilic is the transmembrane protein. Okay, transmembrane protein, or we call it as the integrin. Okay, so transmembrane protein or the integrins, proteins that are embedded in the plasma membrane, are also amphiphilic. The protein have both hydrophobic and hydrophilic region. Okay, so if you look at here, okay, in this diagram, this is the phospholipid bilayer. Okay, so if in the question, for example, it asks you to label the phospholipid bilayer, okay, so you have to highlight from the head of phospholipid molecule for one of the layer until, okay, you cover the head of the other layer, okay, of the phospholipid molecule. Okay, so that is phospholipid bilayer. Do not just simply, okay, tarik macam ni, label phospholipid bilayer. Okay, so you have to exactly point out which is the region of the phospholipid bilayer. If the question asks you to label the hydrophilic heads, then you specifically point at the hydrophilic head. Same goes to the hydrophobic tail. Okay, so if the question asks you to point at the hydrophobic tail, you point exactly at the hydrophobic tail. So please, take note of the questions. Pay attention to what the question wants. Okay. All right. Okay, so 3.2, structure of plasma membrane. Okay. So this the structure of plasma membrane, it follows a model. We call it as fluid mosaic model. Okay, this model is proposed by these two scientists, which is as Jonathan Singer and Nicholson. Okay, so which is proposed in 1972. So far, you don't have to remember the year, but you have to remember the scientists. Okay, Sanger and Nicholson. You have to remember. You have to know. Okay, and then these two scientists propose the fluid mosaic model. Okay, so in this model, it states that the membrane is a mosaic of proteins embedded in the fluid bilayer. Okay. Sekejap. Never mind. Okay, saya nak hilangkan tadi. Okay, so hilang ini, hilang tak apalah. Okay, so why is it the model called as the fluid mosaic model? So if you look at the definition, you have to know the definition why does this model called as fluid mosaic model. Okay, fluid, it is because the plasma membrane, okay, it is not static. The plasma membrane, they can move. Okay, they can move. So there are for the phospholipid molecule, okay, the two layer of the phospholipid molecule, one of the movement of the phospholipid molecule in the plasma membrane, they can move through a movement, we call it as lateral movement. Okay, dia akan bergerak secara mendatar macam tu kan, kiri kanan, macam alunan air macam tu lah kan, kiri kanan macam tu. Okay, lateral movement. And another movement, we call it as flip-flop. Okay, flip-flop. So the molecule at on the top layer can and the bottom layer here, they can switch place. Okay, so they can switch place. That is what we call it as flip-flop. So that's why the model is called as fluid. Okay, the molecule, the plasma membrane is not static. It can move. Why does it? Why does the plasma membrane cannot be static? Because if the plasma membrane were to be static, frozen solid, then the cell cannot function. Okay, the cell cannot function. And then the protein that are embedded in the plasma membrane also can move, but the protein that is embedded will move laterally only. Kan, dia tak boleh nak flip flop. Okay, so lateral movement for the transmembrane protein. So why does the model call as mosaic? Because you have other molecule, which is protein, for example, that is embedded in the plasma membrane. So if you look at from above, okay, so it seems like the arrangement of protein that are embedded in the plasma membrane, it seems like the arrangement of mosaic tiles. Okay, macam tu. Okay, so so that is why the model is called as fluid mosaic model. Okay. Okay, so this is a diagram, okay, from Campbell. Okay, so it shows you the plasma membrane. Okay, the components of the plasma membrane. So as you can see, it has the phospholipid bilayer. Okay, and then you have the transmembrane protein. Okay, transmembrane protein or integrins or integral protein. Boleh panggil dia banyak, tiga lah. For the protein that is embedded, you can call it as integral protein, okay, transmembrane protein or integrins. Okay, so these are the protein that is embedded in the plasma membrane in the phospholipid bilayer, and it is amphiphatic. Why is it amphiphatic? Because it has both hydrophilic and hydrophobic region. So if you look at the diagram here, so this is the region of the integral protein that is hydrophobic. Okay, because it is in the core of the phospholipid bilayer. Dia tertanam pada bahagian dalam that is isolated from the aqueous surrounding, aqueous. So inside you have the cytoplasm. Okay, so this is the cytoplasmic site. Okay, plus cytoplasmic site. Outside you have the extracellular fluid. Okay, extracellular fluid. So both sides are aqueous. Okay, are aqueous. So because it is aqueous, so the region that is not embedded, okay, they are hydrophilic. Okay, same goes to the head of the phospholipid molecule. Okay, so phospholipid molecule is also amphipathic, has both hydrophobic and hydrophilic region. Hydrophobic dalam core ni, okay, which is isolated from the aqueous solution. So another molecule that are embedded, okay, that are found embedded in the plasma phospholipid bilayer is the cholesterol. Okay, so cholesterol. So remember, cholesterol are only found in the plasma membrane of animal cell. Please take note of that. So cholesterol will not be found in the plasma membrane of plant cell. Okay, there is no cholesterol in plant cell, in the membrane of plant cell. Okay, so and then another component of the plasma membrane, you have this molecule, we call it as glycolipid. What is glycolipid? Glycolipid is where short chains, okay, short chains, this is short chains of carbohydrate. Kita dah tengok dah masa chapter 1 hari tu, under carbohydrate, which is carbohydrate, you can classify them as monosaccharides, disaccharide. The third one is oligosaccharide before it goes into the polysaccharide. So the third group, which is oligosaccharide, short chains of monosaccharides that are joined together. Okay, so the chains are consist of a few monomers of carbohydrate, a few glucose strands. Okay, so the oligosaccharides, small chains of carbohydrates that are joined or linked to the phospholipid molecule on the plasma membrane, we call it as the glycolipid. And then the other one is the oligosaccharides can also attach to the integral protein. Okay, so this we call it as the glycoprotein. Okay, glycoprotein. So glycoprotein are short chains of carbohydrate that is attached to the protein that is embedded in the plasma membrane. And you have to take note that when you look at this diagram, the branch of the carbohydrate will always face outside the cell. It always face outside of the cell. It always face the extracellular fluid. If you were to draw the chain facing inside, okay, your diagram will be wrong. Okay, so the carbohydrate will always face outside because it has specific function that we are going to look at later for the role of membrane protein. Dia ada fungsi dia. Kenapa dia kena terletak kat luar? Okay. And then another component of the plasma membrane is you have the peripheral protein. So peripheral proteins are protein which is not embedded in the plasma membrane. They are the protein are found attached to the plasma membrane. Okay, on the inside of the cell. Okay, the cytoplasmic site. Okay, so in peripheral, kat tepi. Peripheral kan bahagian tepi-tepi. Okay, so peripheral proteins are found attached, okay, to the plasma membrane on the cytoplasmic site. Okay, so the peripheral protein can also be found attached to the integral protein. Okay, but they have to be in the inside of the cell, cytoplasmic site. Okay. So another thing that you have to take note is that, okay, for the cytoplasmic site, you have the microfilaments, okay, or the cytoskeleton. So the microfilaments, okay, will attach to the integral protein or it attached to the peripheral protein here, okay. So the attachment of the microfilament of the cytoskeleton to these proteins is to enhance or is to hold the structure of the plasma membrane. Kan? So, as you know, okay, so this is the arrangement for animal cell, for example, okay, animal cell. So animal cell does not have any plasma, it does not have any cell wall, okay. So cell wall. So in order to support the plasma membrane structure, okay, that is the function of the cholesterol. The microfilaments. So the microfilaments will attach to the proteins that are found on the plasma membrane. And then on the outside, okay, you have the ECM, okay, the extracellular matrix, which is composed of the collagen, the fibronectin, and also the proteoglycans. Okay, the three components of the ECM. So basically, there's one question, okay, that is asked during the test exam, okay, exam test. The question asks you to draw the structure of plasma membrane. So you are not expected to draw it like this, okay. As I mentioned, you have to draw, your drawing is just a simple drawing, okay. So how to draw the plasma membrane in a more simpler form. So sekejap okay. Tak boleh main. Kejap ya. Okay, so if you were to draw the plasma membrane in a more simpler form, you just draw it. Okay, you have the phospholipid bilayer. Okay. Okay, so you draw the head of the phospholipid molecule, so like that. Okay, bila guna pen, it will be more easier. So I'm using my fingers, so it is harder to get the picture nice. Okay, so you for the tail, please remember that for the each of the phospholipid molecule, it has two tails, not one but two. Okay, if you draw it as one tail like that, okay, so it seems that the membrane is composed of sperm cell. Macam tu, sperm cell, you have the head, you have the tail. Okay, so make sure you draw the phospholipid molecule of the phospholipid bilayer. Each one will have two tail. Okay, so that is that is phospholipid molecule. Okay, not sperm cell. Okay, so make sure. Saya mention ni sebab bila dalam exam dulu, pernah ada dia draw only one tail. Okay, so that will be wrong. Okay, your structure for the phospholipid bilayer will be wrong. Okay, so and then you have the transmembrane protein. Okay, transmembrane protein embedded. Okay, so you label, okay, inside you have the cytoplasmic site. On the outside you have the extracellular fluid. Okay, so you can have carbohydrates attached to the protein, so you can label this as what? Glycoprotein. Okay, you can have apa nama? Okay, carbohydrate that is also attached to the phospholipid molecule. See, this one, kena tak ambil the whole thing, okay, is the glycolipid. Okay, glycolipid. And then you have another molecule, we call it as the cholesterol. Okay, if you look at the original diagram there, cholesterol are only drawn on one of the layers. Okay, and it is embedded inside. So cholesterol, it consists of 4 fused string. Kita dah belajar chapter 1, okay? 4 fused string, 1, 2, 3, 4. 4 fused string, and you draw it on one of the layer, okay, of the phospholipid bilayer, okay? Biar tengok kat sini kan. So the cholesterol is embedded in one of the layer, and it consists of 4 rings that are fused together. So please take note. Okay. So if you were to draw it, for example, the cholesterol molecule, okay, in between the bilayers, okay, so that would be wrong. If you draw the cholesterol protruding, okay, from the membrane, so your cholesterol will be wrong. So you are going to lose marks. Okay, so make sure you take note of that. Okay, so on the complete kan lah kan. So and then so this is the integral protein, and then you have on the inside the peripheral protein. Okay, so always remember to draw the peripheral protein on the inside of the cell. Glycoprotein, glycolipid facing outside, and then you have collagen, okay, and then you have the fibronectin. Okay, fibronectin will attach the integrins, okay, to the collagen, okay, and then you have network of proteoglycans, okay. So draw it neatly lah. This is my just a rough drawing, okay, of the plasma membrane. So just draw it simple but deep, okay, not like mine. So dia just a sketch, okay. So so that is for the plasma membrane. So you have to label all the components and draw it properly. Okay, so next is the still on the structure of plasma membrane. So now we are going to look at the fluidity of the membrane. So according to model tadi, the plasma membrane, it follows the fluid mosaic model. Okay, so you know why is it mosaic? You have, because in the plasma membrane there is other molecules that are embedded. You have protein, you have cholesterol, you have glycolipid, you have glycoproteins. Okay, so that's why it is called as mosaic. So fluid, okay, because the membrane is not static. Okay, the membrane is not static. So there are 3 factors that affects the membrane fluidity. The first one is the movement of phospholipid molecules. So I said the phospholipid molecule will have 2 types of movement, which is lateral movement and also flip-flop. Okay, flip-flop. And then the protein can also move through lateral movement. Okay, but the movement of the protein will be slower compared to phospholipid molecule, because phospholipid molecule is the size is smaller compared to protein. The size of the protein is large. Okay, so the movement of protein will be much slower compared to the phospholipid molecule. And then the presence of unsaturated fatty acids. Okay, so this is the presence of unsaturated fatty acid on the phospholipid molecule. Okay, so you know that the phospholipid molecule consists of the head and the tail. Okay, the head and the tail. For example, for plant cell, okay, the phospholipid molecule, okay, the tail, okay, can have the hydrocarbon tail, the fatty acid can be bent. Okay, it can be bent. Bengkok. Okay, why is it bent? Because here the carbon atoms, okay, on the bent region has double bond. Okay, so that indicates this the hydrophobic tail is actually made up of unsaturated fatty acid, where the carbon has double bond and causing the molecule to be bent, or we call it as kink. Okay, so this one, it applies for plant cell. Okay, it does not apply to animal cell. Okay, it applies to plant cell. Okay, so the presence of cholesterol applies for animal cell only. So kena tahu presence of unsaturated fatty acid is for plant cell. Okay, to cause the plant cell, the membrane of the plant cell to be in the fluid state. Okay, it prevents the membrane of plant cell to be static, to be frozen solid. Okay, and as for animal cell, okay, how to maintain the fluidity of the membrane through the presence of cholesterol. Okay. Okay, so the first one, movement of phospholipid molecule. Okay, for movement of phospholipid molecule. So uh, they are held together by hydrophobic interactions. Okay, the phospholipid molecule in the phospholipid bilayer. Okay, they are held together by hydrophobic interaction because in the core of the plasma membrane consists of the tail. Okay, the tail of the phospholipid molecules that are interacting to one another through hydrophobic interaction. Okay, and then the phospholipid molecule carry out flip-flop movement, okay, across the two layers of the phospholipid. So flip-flop, one phospholipid molecule on the top, it can swap place with the phospholipid molecule at the bottom layer. Okay, they swap places. That is flip-flop. So the lateral movement of phospholipid within the same layer of the phospholipid. So lateral, kiri kanan lah, kan, dia bergerak kiri kanan macam tu. Okay, some protein can also move laterally, but the movement of protein compared to phospholipid, the movement will be much slower. Okay. Okay, so for the movement of the phospholipid molecule, the lateral movement is more frequent. Okay, frequent. Okay, about 10 to the power of 7 times per second. Oh, imagine lah, kan, berapa banyak pergerakan tu dalam satu saat. Okay, so tapi for flip-flop, the movement is rare. Okay, it rarely occurs about once per month. Okay. Okay, so the presence of unsaturated fatty acid will only occurs in plant cell. Okay, plant cell. So as temperature cools, membrane switch from a solid state, from fluid state to solid state. So solid, it will cause the plasma membrane to be static, kan? Static. So if static, the plasma membrane cannot carry out its function. For example, to allow the entry of molecule in and out of the cell. So this can cause the membrane to not be able to function properly. If the membrane cannot function properly, the cell will not function. So what happened when for example, can you have a plant, okay? So plant that are grown in cold temperature, okay, during winter, for example. So how to prevent the cell of the plant from being frozen solid, okay, and cause the plant to not be able to function, okay? So in order to prevent the cell from frozen solid and not function, so the cell will increase, okay, the amount of unsaturated fatty acid of the phospholipid molecule in the plasma membrane. Okay, it increases the percentage, okay, the percentage of unsaturated fatty acid of phospholipid molecule in the plasma membrane. Okay, so this makes sure, okay, kalau kita dah belajar kan, kalau the molecule is bent, okay, the molecule of plasma membrane is bent, so this prevent the phospholipid molecule from okay, the phospholipid molecule from closely packed together and prevent the cell from the plasma membrane, okay, from turning solid, okay, it prevent the cell from being frozen, okay. So this makes sure that the plasma membrane maintain in fluid state, okay. So that's why for plant cell, the hydrophobic tail, okay, of the phospholipid molecule, it increase the amount of unsaturated fatty acid. So the temperature at which a membrane solidifies depends on the type of lipid it is made up of, okay. So it says here, the membrane rich in phospholipid with unsaturated hydrocarbon tails, okay, which is the presence of unsaturated fatty acid, the molecule is bent, okay, due to the presence of double bond between the carbon. so will remain fluid even the temperature drops, okay, at lower temperature. So unsaturated hydrocarbon tails cannot pack together, okay, they cannot pack together because the molecule, okay, the tail is bent, okay, as closely as saturated hydrocarbon tails due to the presence of a double bond between the carbon to form kink. Okay, so kink in the tail, so the membrane will be more fluid. Okay, so imagine kalau dia punya tail straight, so the phospholipid molecule, if the temperature drops, okay, so the membrane, the phospholipid molecule can closely pack together and become solid, okay, at cold temperature. So the cell cannot function. Okay, the cell cannot function. The plasma membrane cannot carry out its activity. Okay. So if you look at this diagram, the comparison between plasma membrane that is made up of all saturated fats. Okay, so if the temperature drops, okay, so the molecule will be closely packed together and causes the membrane to be solid and not functional. Okay, but if the membrane increases in the composition of its unsaturated fatty acid, okay, so the tail will be bent. Okay, so this will prevent the molecule from closely packed together and maintain fluid, okay, even if the temperature drops. Okay, so unsaturated hydrocarbon tails of phospholipids have kink that keeps the molecule from packing together and membrane will be more fluid. Okay, so membrane can carry out its function. Okay. Okay, so it says here the lipid composition of cell membranes can change as an adjustment to changing temperature. Okay, so the lipid composition, the fatty acid, okay, fatty acid. So it can increase the amount of unsaturated fatty acid of the phospholipid molecule. For example, in plants that tolerate extreme cold temperatures such as winter wheat. Okay. So the percentage of unsaturated phospholipid will increase. Okay, unsaturated fatty acid of phospholipid of the plasma membrane for this plant. Okay, winter wheat. So will increase in the autumn. Okay, in the autumn before the season change to winter. Okay, so that keeps the membrane from solidifying during winter. So autumn adalah musim luruh sebelum dia masuk ke winter. Okay, which is snow, right? Okay, so next is the presence of cholesterol. Okay. For the presence of cholesterol, okay, so cholesterol, you have to remember it is only found in animals. Okay, they are not found in plant cell. Okay, plant cell tak ada, ada dalam animal sahaja. Okay, so the cholesterol has different effects on membrane fluidity at different temperature. So the cholesterol is wedged, okay, between phospholipid in plasma membrane of animal cells. So please highlight, they are found only in animal cell. Okay, so wedged means that they are found in between the phospholipid molecule, okay, on one of, on both layers lah, okay, of the phospholipid. So kalau nak draw dia, awak draw dia on one of the layer, bukan in between or protruding out. Okay, so they are found wedged between the phospholipid molecule in the plasma membrane. So the function of the cholesterol will be different at different temperature. Okay, the function of the cholesterol will be different at different condition of temperature. So for example, if the temperature is warm, your body, okay, has to be maintained at warm temperature, which is around 37, for example. This is the ideal temperature of your body for enzyme to work. Okay, so as you know, your plasma membrane contains protein that function also as enzymes. Okay, so cholesterol at warm temperature, the function of the cholesterol is to restrain, okay, restrain the movement of phospholipid and the membrane become less fluid. Biasalah kan, kalau macam contohnya, if molecules, okay, you increase the temperature, you imagine the molecule, the movement of the molecule will be very fast. Dia akan ada tendensi untuk bergerak dengan cepat. Faham? Sama juga kalau phospholipid molecule dekat plasma membrane, okay, if the temperature is warm, okay, so this causes the phospholipid molecule to be so fluid. Okay, the movement of the phospholipid molecule will be very fast. Kan? So the membrane can even lose its structure. Okay, so in order to restrain, okay, kurangkan pergerakan fosfolipid tu, kena ada kolesterol in between, kan? Dia macam bertindak sebagai penghadang lah, kan? So penghadang dekat untuk menghalang fosfolipid tu daripada bergerak terlalu laju at warm temperature. Okay, so that is the function of cholesterol at warm temperature. So what is the function of cholesterol at cold temperature? Okay, when the temperature drops, cool temperature. So when the temperature drops, okay, for plasma membrane of animal cell, dia tak ada unsaturated fatty acid, dia tak akan ada kink, kan? Dia tak akan ada kink. So when the temperature drops, so the phospholipid molecule will have the tendency to closely pack together and become solid. So by having cholesterol, okay, embedded or wedged between the phospholipid molecule there, so this prevent the phospholipid molecule from closely packed together and prevent the phospholipid molecule, okay, from closely packed together and prevent the plasma membrane from becoming solid. Kan? So imagine kalau dia hanya ada saturated fat. Kalau animal cell, dia punya hydrophobic tail dia only saturated fat. So imagine if the temperature drops, kan, kalau duduk kat tempat yang sejuk contohnya, sel awak akan membeku, kan? Sel awak akan membeku. So untuk prevent yourself from being frozen solid, so that is the function of the cholesterol that is wedged between the phospholipid molecule, which is to prevent the phospholipid molecule from closely packed together and slow down solidification of the membrane. So remember, membrane cannot solidify. If it solidifies, it cannot carry out its function. Okay, so this is a diagram to show you the presence of cholesterol in the membrane. Okay, so basically the presence of cholesterol is to maintain the fluidity of the membrane. Itu secara general. Tapi bila soalan dia tanya what is the function of cholesterol when the temperature drops, for example at cool temperature, so prevent close packing of phospholipid molecule and prevent the plasma membrane from solidifying. Kalau soalan tu ditanya what is the role of cholesterol when the temperature increase, okay, so this is to restrain the movement of phospholipid molecule, okay, from preventing the membrane to become too fluid, okay. So this is to maintain the fluidity of the membrane just right, okay, for warm temperature, okay. So cholesterol, it says here, cholesterol reduce membrane fluidity at moderate temperature, moderate. Tapi kalau dah increase pada 100, so tak boleh nak buat apa lah dah, kan. Memang plasma membrane tu akan terlerai lah. Okay, but at moderate temperature, the presence of cholesterol is to reduce fluidity, okay, because it restrains the movement of phospholipid molecule, okay, reducing the phospholipid movement. But at low temperature, it hinders, okay, boleh guna perkataan hinders, prevent, okay, solidification, okay, by disrupting the regular packing of phospholipid, okay. So it prevents the membrane from solidifying, becoming solid, okay. Okay, so cholesterol will act, so therefore, okay, cholesterol will act as fluidity buffer, okay, fluidity buffer for the membrane. So okay, sekejap ya. Okay, Okay, so ada background music Didi kejap. Okay, so kita sambung. Cholesterol acts as a fluidity buffer for the membrane. So it resists changes in membrane fluidity caused by changes in temperature. So to sum up, okay, the role of cholesterol, basically it functions as fluidity buffer. Okay, it controls the fluidity of the membrane from becoming too fluid, from becoming too solid. Okay, that is fluidity buffer. Okay, so membrane must be fluid to work properly. So when the membrane solidifies, its permeability change and enzymatic proteins in the membrane may become inactive. Okay, so that's why membrane cannot be too fluid or turn solid. Okay. Summative question, these three components of plasma membrane. This one you please do on your own. Okay, please three components of plasma membrane. Ini untuk punya revision lah. Okay. So name two types of phospholipid movement. Okay. Number 3, how unsaturated hydrocarbon tails membrane fluidity. Okay. So number 3, remember it only applies for plant cell. Okay. So next is membrane function and membrane protein and their function. Sekejap. So it is nearly 1 o'clock. So I guess we stop here for now. So do you have